challenge in environmental epidemiology, and that's the use of omics data. Omics has pretty much revolutionized medical research over the last few years, even leading to things like personalized medicine. And it's now becoming really important in environmental research also. And it covers everything from potentially developing internal dosimeters that you might see to mechanisms to early indicators of disease. And I think ROEL is going to focus in particular on the dosimeter aspect. How can we identify internal markers that may tell us something about the exposure that people examine? So go ahead.
Thank you very much for the kind introduction. Also want to extend my thanks to the organizers to invite me here. Francesco put it at the opening to this party, and really has been a wonderful conference, and so I've really enjoyed it, so thanks for having me here. Before I start my talk, I just want to say that I have no conflicts of interest. So what I'm going to talk about today is how omics technologies can be used in environmental health in trying to delineate basically the relationship between exposure that we have and how they actually may be related to detrimental effects on health or positive effects on health. If we reflect on the theme of this conference, which is titled Old and New Risks and Challenges for Environmental Epidemiology, then the first question that comes to mind basically is, what do we know at this moment really about old and new risks? And this is taken from the Franzafar paper in 2015 on the Global Burden of Disease project in 2013, where it's listed basically what is the attributable proportion of environmental factors, behavioral factors, and metabolic factors, in which we can see in the pie chart in the middle that potentially we can explain about 40% of the attributable proportion. Of which environmental factors, either alone or in combination with behavioral and metabolic factors, may explain about 30%. However, it's also clear from the gray circle around it that there's still a large part of this pie that we actually cannot explain at this moment. And by extension, given that we know that environment is important of the things that we do know, and assuming that we didn't do better than some of the other fields, we can think that indeed in the gray part there's still a lot of environmental factors to be discovered. So what I want to address today is basically how can omics technology try and help in elucidating this unattributable proportion of this pie. But before I do that, I just want to do a disclaimer that there's not one road that leads to Rome. There are many roads that lead to Rome, and we have heard wonderful presentation in this conference about how satellite information, sensors, mobile devices can potentially be used to improve exposure assessment and therefore try to get a clearer picture how exposures may relate to diseases. So if we go back and think how molecular markers and by extension omic marker can really help in studies of environmental health, then we basically can distinguish about 4 areas. One is that we could use these markers to enhance exposure assessment. Classical biomonitoring of lead is of course a classical example. It can complement studies of disease etiology by evaluating the biological plausibility and the mechanisms of suspected environmental hazards, and therefore may aid in the risk identification and in some way add to the proof that something is related to an effect. It can aid in a more refined derivation of the exposure response association. And if we think about old risks, a lot of the questions that we still have is about what is the exact shape of the dose response curve, are there functional thresholds, are there susceptible populations? These questions are still out there. And we can potentially use these markers if we would have markers that predict for disease to actually find exposure that may have a potential disease risk even before the disease occurs. Of course, that's what we would like to do. So in the present today, I will basically try to focus on 3 of these questions. So as was said in the introduction, omics markers can be used in environmental epidemiology basically through the whole cascade from exposure on the left side to disease on the other side. And we basically can use it for improving exposure assessment, but also for a better classification of disease on a molecular level, which may give us a clear picture as well. But indeed, for the talk of today, I will basically focus on what we now term the field of exposomics, where we try to use these omics technologies to get a better handle or a better assessment of what the exposures are that may be related to disease and how they actually may either directly be measured or by their biological response. I will not go into translational research today. So what I will do in this talk, I will just go through some of these elements, and I will use some proof of principle studies to illustrate how this actually may work. So the first example is basically the question, can we use omics markers to basically enhance exposure assessment by trying to directly measure the exposures through omics technologies? And technologies that come to mind that are basically currently here and are being developed is metabolics, which basically helps to screen for all small molecules by high resolution mass spec, or adaptomics, which tries to measure the whole complement of all protein edits. What I show you here is an example of how we have used metabolomics or small molecule screening in an occupational setting of people exposed to trichloroethylene, which is an occupational exposure because of degreasing activities, but it's also an important environmental exposure as a soil and water contaminant. And this is a study that is led by Qing Lan of the National Cancer Institute, and the material that I'm going to show is in a paper that's now in press by Dr. Walker, who is here from Emory University. So what we did was we basically enrolled 80 workers exposed to high levels of trichloroethylene, 96 healthy controls, collected plasma, and basically ran metabolomics on these plasma samples. In this slide, I show you the result of that experiment. So what you see basically here are all these small dots, which are basically 10,000 features that are measured in the plasma, of which about 200 basically are under an FDR control of 20% related to TCE exposure. The ones in red here are positively correlated to the TCE. The ones in blue are negatively correlated to TCE. Now, what you can take away from this picture is that actually in this left corner, we have this stack of metabolites that are highly significantly perturbated and that are highly correlated to the TCE exposure. And so if we look further, and this is the work done by Dean Jones and Douglas Walker, we're able to basically map those features to what those are. And we're able to show that here these are metabolites of TCE, trichloroacetic acid, and trichloroethanol, which were also confirmed by looking at and correlating them to the urinary TCE exposure markers. So we're able to find in the plasma indeed directly metabolites of exogenous exposures. Hence, this is an exposure situation which is extremely high and may not translate directly to environment, but given that metabolomics and high-resolution mass spec is developing so quickly, this leads to the opportunity to actually do that. But what is more exciting is actually that we identified here all these green arrows, which are metabolites of TCE, because they are actually halogenated, but of which we actually don't know what they are. So with actually doing this agnostic approach, we identified a bunch of new metabolites that we had never observed before. And why is that interesting? Well, because of this. If we actually take that one step further, we then related the measured metabolites and measured exposure directly in the plasma to previously measured biomarkers of effect. And so this is basically looking at nephrotoxicity markers, especially KIM1 and NAG, which was published in 2012. And what you can see and may appreciate is that actually the relationships between these effects are all done by the unidentified metabolites. It's none of the known metabolites. It's actually completely driven by all the unknown metabolites. And so we would have never known this if we would not have used an agnostic approach to actually make the link between the phenotypical expression and what the exposure was. Similarly, immunological markers, which were previously published by Lanadal also in 2012, has exactly the same pattern. Here we have the unidentified metabolites, which are related to the immune markers that we had found previously to be perturbated in these workers. So there's actually two take-home messages from this example. One is that indeed this technology, if sensitive enough, can be used to actually screen widely for features which includes exogenous exposures. Secondly, it can help us actually to make relationships between the exposure and between metabolites that we didn't know before or even didn't know they existed to actually the health effects that we observe. So there's another approach that one could take, and that is not by actually trying to directly measure the exposure as you do in metabolomics or in adaptomics, but it's actually to try to measure the imprint that certain exposures may have in the biological system. And may that be an exposure-specific signal, or that may be a broader signal that actually has a biological imprint, or what could be termed as an allostatic load. And so we can do that by trying to use epigenetics, transcriptomics, or microbiomics. So one of the more clear work that has been done is basically on smoking-induced changes in methylation. So I'm going to use that throughout the presentation kind of as an example of how this can be used. Again, it's a high exposure situation, but it shows what these technologies potentially could give us. So this is based on the publication by Jobert et al. In 2016 of the PAGE consortium, led by Stephanie London of the NHS. And what you can see is that maternal smoking in pregnancy is related to perturbations in the methylome, where you can see here, oh, sorry, that's the wrong button. The different chromosomes and the different CpG islands on the chromosomes, and you can see that there's about 6,000 of these CpG sites, methylation sites, that are differentially methylated on an FDR control, and there's about 600 under a more strict Bonferroni control. So indeed, smoking leaves some kind of imprint in the methylome of newborns. And most significantly are the HAR receptor and the myosin 1G CpG site. So that's interesting, and it also confirms to what we observed previously also in adult studies. So this is actually looking at adults, smokers versus non-smokers. Do we actually see changes in the methylome? And so what you can see here is indeed that we see hypermethylation, again, of the HSR receptor, and that we actually see myosin 1G as well here to be hypermethylated, similar to what's observed in the PAGE consortium, here observed in adults. So very consistent observations, which now have been done in many, many studies. What was a different question that was asked, and this is a paper by Florence Guida under supervision of Paolo Venice and Marc Jadot, is to actually do the analysis in a slightly different way. And so the question that we were asking to the data is, well, we have this imprint, but can we use this imprint to actually see if it tells us something about the life course of the exposure? Is there something true that it's not an immediate change or a change that is there but actually may return back? Or can we actually see that there is indeed this long-lasting imprint? And so what we did in this analysis is basically recoding former smokers to current smokers based on the time since quitting smoking, so that in every single tier of the analysis we have the same power, and then we basically move the slider out of time since quitting smoking. And what you can see here of all the CpG islands that are perturbated in one of these analyses, which are about 700, when we start moving out, and basically time since quitting, you can see that some of these CpG islands seem to revert back to normal, so they're reversible. Where there are some CpG islands that actually remain to be differentially methylated even long after the exposure apparently has gone away. And of course, this is not on individual data, this is basically on a population level derived. If this is true, this actually gives us the idea that we may be able to actually use these imprints to actually measure of exposure and even have a look and see in the history of the exposure of people. This was not the first observation about reversible and irreversible. What I show you here is a paper that was published by Bean et al. In Genome-Biology in 2007, looking at reversible and irreversible effects again of tobacco smoke in the airway epithelial gene expression. And similar to what I showed in the methylation results, what you can see here is that again, when we look at non-smokers versus former smokers and current smokers, that the former smokers and current smokers look alike in gene expression, but are different than the non-smokers. And so these would be irreversible changes. In this panel here, what we can see here is that actually the non-smokers coincide with the former smokers, but are different than the smokers. And so these are reversible genetic changes. And so what we then can ask is basically the question, well, that's interesting that we have these histories and we see things changing, and some of these changes may actually be long-lasting. So even if the exposure has gone away, we may be able to still detect that difference that has occurred in the past. Well, are these specific to tobacco smoke? Are we looking at a reflection of exposure? Is this biological response? Is this an early effect, or is this basically a disease manifestation? And so I'm going to go into the next set of slides to try to tease out the answer to these questions. So in a follow-up study in an area in China, where we're conducting a long-term study on lung cancer, which is led by Qing Lan, where there is an increased lung cancer rate, which is approximately 3 times higher than the Chinese average, with even higher hotspots in this area. From all the research that has been done in this area, we know that this increase in lung cancer risk is associated with the use of a specific bituminous coal in that area, which is termed smoky coal, if we contrast that to smokeless coal, which is also used in the area, which is an anthracite coal. So we're able to look together with the lab of Evispira in Boston University at the gene expression of buccal epithelial cells, in this case, of women living in Jiangwei that are exposed to these high levels of house or air pollution. And ask basically the same question: do we see a difference in expression profile when we compare the women that actually are having an exposure to the high-risk coal versus the women that actually have an exposure to the low-risk coal? And what you can see here is basically what we term here cluster 1, which basically is largely underexpressed in the high-risk group compared to the smokeless coal group, where we have cluster 2, which is an overexpression compared to the low-risk group. That's interesting, and then we asked a more nuanced question, and the question that we asked is basically, well, if we now actually take what we observe in China in buccal epithelial cells, and we overlay that to the study that was done at Boston University among smokers, and we combine those two genetic profiles, what do we see? And so this is a gene set enrichment analysis, where you can see here the gene enrichment score, and we can see here these are the cluster 2 genes which were overexpressed in the high-risk group. And what we can see here, if you have here the smokers, then actually you see gene set enrichment here. The genes are more enriched also among smokers, among the genes that we actually found among the women that were exposed to high levels of house or air pollution. Similarly, if we look at cluster 1, which were lower expressed in the high-risk group, similarly we see gene set enrichment, which are indicated by the bars here. The higher, the more the gene is enriched among the smokers as well. So we see significant overlap in the response that we see among smokers as what we see actually in the response that we see among household air pollution. Now, recently we had the opportunity to do these assays as well among people that are exposed to carbon nanotubes. And carbon nanotubes, again, a respiratory particulate that we think may be related to detrimental health of the lungs and potentially to lung cancer. And so we asked the same question: if we actually look at nasal epithelial cells here, again a proxy of the respiratory tract, and we look at gene expression, what do we see? And in this study where we compared people exposed to multi-wall carbon nanotubes and unexposed, we basically observed the following. And so this is basically going to question 4 that I posed, where the other ones may actually add to the biology and the relationship that we observe. Here the question is basically, well, if those patterns are there, can we now actually take an exposure of which we actually know very little and actually understand what that exposure pattern or that transcript pattern may mean? So that's what I show you in this slide, and what we observe again, this is in gene set enrichment analysis. So we took the genes that were differentially between people exposed to carbon nanotubes compared to the controls. And what you can see here, similarly with a gene set enrichment analysis, that there is this suggestion again that the effect in smokers seems to be similar to what we observe in multi-carbon nanotubes, exactly the same that the ones that are underexpressed in carbon nanotubes seem to be somewhat enriched in smokers as well. This is a small study, so it didn't reach statistical significance, but it shows promise that if we actually start to understand these patterns, that we actually could apply them to different exposures and learn what their potential biological and detrimental health effects may be. We did a similar study, and this is by Nahid Mostafazi, published in Epidemiology, accepted in Epidemiology, where in this case we looked at air pollution and genome-wide gene expression in blood samples. And so you here see a set of genes that were differentially expressed with NO2 exposure, and this study was done in Epic, Italy, where we have relatively high NO2 concentrations. And you can see again here that we have several genes to be differentially expressed with NO2 exposure. And again, similar to the other results that I showed you, we asked again the same question. If we see this pattern, how does it actually compare to what has been described as the gene expression-based signature of smoking status published by Beneke et al. Based on the PREDICT consortium? So again, took that profile and compared that to what we actually observe here among the people exposed to higher levels of air pollution. And similarly, although much less impressive, we don't see any gene set enrichment among the genes that were positively associated, but among the genes that were negatively associated, so suppressed, we see a marginal effect of gene set enrichment, where you can see that the distribution is slightly lopsided to the left side with a significant p-value. So there might again be some reason to assume here that what we observe there has some overlap with what we see with smoking. So if you put that all together in some kind of graphical presentation, then this is basically the picture that starts to emerge. We basically have, if we look at the respiratory tract carbon nanotubes, which showed a differential expression, we have the household air pollution, which all overlap in some way with smoking, but may also be different. If we look at the blood gene expression, then we see that the outdoor air pollution that I just showed you has some marginal overlap with smoking as well. And so when we go back to the question that I asked when we're talking about irreversible and reversible changes, then at this time we're not sure where we are exactly, but there's some evidence that these exposure profiles or these imprints that we see either in the epigenome or in the transcriptome may be not completely exposure specific and may reflect some biological pathway. So this is still developing work together with the National Cancer Institute. We're doing multiple other studies on particulates exposures to build up this database to see in how much do we actually have here, which I put here in a simple 2x2 table, what is the evidence that indeed these exposures leave some kind of specific imprint in the biological system, which maybe if it's no, of course, then that makes these imprints less actionable. We don't know where it's coming from. It might be multiple sources, but of course still may be interesting to understand the biology of the disease. However, if it would be specific to exposure, that actually makes it actionable as well. We know what is actually driving the risk, and if we actually can relate that directly to disease risk or to the biology, then that would be very informative markers. What I showed you until now is all based on cross-sectional studies. And of course, what we really would like to do is to, in one single step, actually make the connection between the exposure, biological perturbations, and the disease risk that starts to occur, which basically has been termed by some the meet in the middle approach, where we have a biological sample which we relate to the disease risk and which we also relate to the exposure, or we understand the signature that we can relate to exposure. And so this, of course, can be done in prospective studies where we have biobank materials that we can use in a nested case control setting where we select, for instance, lung cancer cases and controls, look at biomarkers that actually may be predictive for disease, and see if those biomarkers are also related to exposures, or we can tease out what the relationship is between those markers and the exposure. Again, I'm going back to a smoking example because I used that in this presentation. This is a paper published by Fasinelli et al. 2015 from the UHF Institute here in Italy. This is work by Paolo Venice, where they showed in 4 different cohorts the main effect of HSRR. And if you remember from the first slide that there was one of these genes that was related to smoking. Similarly, the F2RL3 gene, which is posted here, depicted here, is related to smoking as well. These are analyses which are matched on smoking. So here it's basically looking at the main effect of these genes on the disease risk. And you can see in the 4 cohorts that they actually are related to the lung cancer risk. They're both all hypomethylated in these studies. If we look at the mediation analysis that was done in one of these studies where that was possible, where it was not matched but basically was controlled for, we can see that smoking through the methylation chains has a mediated effect of about 30% and together about 37%. So what this shows is that indeed if we actually can use these prospective studies to actually link these profiles to disease risk, that may be able to actually make a better connection between the environmental factors that actually cause disease risk. So to go back to the main question that I posed, is can exposomics aid in delineation from exposure to disease? My answer would be, with these examples, yes, we potentially can. But it's also fair to say that it's still early days. There are still issues with sensitivity and coverage of the analytic platforms. I showed you here examples of exposures that are really high and have a strong toxicological effect. In these settings, we can detect those. That doesn't mean that we can detect them in all different settings. There are issues about exposure or biological specificity, as I showed you with these overlay of these different studies, how they actually overlap or don't overlap. Of course, there's also issues about tissue specificity if you think of biological response. We looked here primarily at genetic changes in the airway, which is also the target organ. But of course, if we start to think about changes to what happened to the brain, and we have blood samples, then of course the biological patterns that you may observe in the blood may not have any predictive value for what actually happens in the brain. And so that is a complication that we have to work through. All this data, which I didn't show, but of course is highly dimensional, which means that statistical and causal interpretation is challenging. And the last point is that although we have many samples biobanked, and I didn't show you an overview, but my estimate is that for adults we have now about 8 million samples biobanked. Although we have many, we don't have many repeated samples, and therefore the chance that a biological sample reflects disease correctly and the exposure correctly, that may be a low chance if we don't really have the systematic long-term imprints that these exposures leave, given that they may actually be reversible and only show up for a short time. That's a limitation that we have. Everything that I showed here are early results and therefore need follow-up. I think they show promise, but we really have to extend this research to get to larger sample size and to really search for replication. That's currently going on in several of the exposome projects, which are funded under the FP7 program, which are listed on the left side, the CHEER program in the US, and the Japanese JAX study. There's a lot of people that I need to thank that basically contributed to many of these samples. Just to highlight Mark Chadeau from Imperial College and Paolo Finis, Jelle Vlaanderen from Utrecht University, and then my former colleagues at the National Cancer Institute, Ching-Lan Rothman and Deborah Silverman. Thank you very much.
We have time for a few questions. Let me ask you a question. You showed some really nice examples where you had access to potentially the target tissue in the lung, but for most epidemiologists, blood is pretty much the tissue that they get. So can you say something from your knowledge about how well or what cautions there should be about people who may be using blood as a marker for exposure when the lung may be the target organ of that chemical?
I mean, that's a difficult question to answer. I mean, we showed basically here indeed target organs, which we are very close to. I showed you the example of air pollution to gene expression, and of course there have been examples also of air pollution and methylation changes in the blood as well. How well those biological phenomena translate to the target tissue is something that I don't know and cannot really speculate on many, but we know that there is real differences in expression profiles and epigenetic profiles if we compare different tissues. If you think about directly measuring the exposures, if you take the metabolomics example where we're not trying to understand it biologically what's happening, but we're just trying to measure the complement of all exposures that are out there, then of course the blood may well be a good approximation of what the internal dose is. That doesn't mean that that is the internal dose at the target organ, but that discussion is not different than how we have used exposure markers in the past, like lead and others, where we have used basically blood proxies as internal dose measures, and that has worked well in many occasions. I think if you go to the biological interpretation, it becomes much more difficult to actually know how well those reflect.
Okay. Joel, you're going to ask a question?
Yeah, it's somewhat related to yours. So in Bob's question, the implication is that it's the methylation in the lung that I care about because that's the target organ, or in the heart or the brain, or depending on whatever outcome you're looking at. But what do you think about the possibility that the way you get from an exposure to, let's say, the brain might be that by changing methylation, you cause white cells to release inflammatory mediators which wind up being at the target tissue, in which case, the blood might then become more relevant. Do you think that that's...
No, that's a very fair point. Of course, it depends on what the mechanism is. Right. And so if the mechanism is localized in an organ, then that likelihood is less. If it's basically secondary to something that happens systemically, and then basically leads to a secondary effect in the target organ, then actually what you observe in the blood may be very relevant. So I think you're completely right. However, for a lot of these things, we don't know exactly what mechanisms are. And I think there's a cautionary note here that this tissue specificity does play a role, and we have to basically try to grapple with that.
Thank you, Roel.
Now, Dr. Shi